wabarakatuh. I am Nabil. Today, me and my group member will be presenting our research on our final year project, including the scope of study therapeutic plant in Malaysia. Our objective is to discuss the effect of therapeutic plant on human health and to observe and describe the morphological characteristic of two medicinal plants that we chose, Andrographis, paniculata, and Melicope telephilia. The main is when you want to identify the bioactive compound present in this plant using qualitative phytochemical test. So our problem is we don't know the composition of the plant, so we will know it through the qualitative phytochemical test. The importance of the study is the research will provide fundamental information on Melicope telephilia chemical content. It also may serve as a baseline for future investigation to its medicinal potential. We want to support the development of natural product within the medicinal pharmaceutical industry. The main method that we use is morphometric study and also phytochemical analysis. So this is the overview of the main result. Andrographis paniculata tests positive for all compound except alkaloid, while Melicope telephilia tests positive for all except terpenoid. Firstly, this is a hypothesis on the research. The plant extract obtained will test positive for saponin, tannin, alkaloid, and flavonoid, terpenoid, phenolic based on standard phytochemical screening outcome from similar medicinal plant. Medicinal plant commonly contain the secondary metabolite, as evident by routine positive result in preliminary screening using this exact reason and procedure.
Hi, I'm not AFIQAH. I'll be talking about the literature review for this experiment. The first sample that we had obtained for this experiment are Andrographis paniculata. It is locally referred as Hampadu Bumi or King of Bitters. This plant belongs to the Acantheraceae family and usually found in China, Taiwan, India, Sri Lanka, Southeast Asia, including the lowland of Peninsular Malaysia. This herb could grow up to 30 to 100 centimeter in disturbed soil and shaded moist forest edges. It is traditionally used to treat fevers, snake bites, and skin conditions such as eczema. Moving on to the next sample is Melicope telefolia. It is referred locally as tenggek burung, and this plant is a member of the Rutaceae family. This plant can also be found throughout the rainforest of Malaysia. This small tree could grow up to 10 meter and prefer the limestone hills and hilly terrain with well-aerated and humus-rich soils. The function of this plant is to ease the digestive problem, reducing inflammation, and heal skin irritation. Moving on to the secondary metabolites that are present in the samples. In Andrographis paniculata, there is saponin, phenolin, tannin, terpenoids, and flavonoids, while alkaloids are generally absent or present in negligible amount. Next is the secondary metabolites in Melicope telefolia, which is saponin, phenolin, tannin, alkaloid, and flavonoid. However, terpenoids are often absent or detected in a low amount. Flavonoid and phenolic compound play an important role in antioxidant activity by scavenging the reactive oxygen species and protecting against the oxidative stress, while coumarin-related compound will contribute to the anti-inflammatory effects through the cyclooxygenase 2 inhibition.
Hi, it's Haneem. For the methodology, we first started with the sample collection. Both samples were purchased from a herb garden here in Kepala Batas, Pulau Pinang, where they underwent two main investigations, which are morphometric study and phytochemical analysis. In morphometric study, the taxonomy of both samples were determined, where the scientific name and common name for each sample was recorded respectively. Next, the dimension consisting of the leaf's length, width, and thickness were measured. Moreover, characteristics of the plant like leaf shape, color, and venation type, as well as the stem color and texture were observed. Last but not least, the stomata density were assessed under the microscope. In phytochemical analysis, the first step is to prepare the sample. Fresh leaves were washed with clean tap water and dried in the oven to eliminate moisture. The dried leaves were then grinded into a fine powder using a blender. Next, the extraction process was carried out using methanol, where powdered sample was soaked in a methanol bath for 2 weeks at room temperature. A vacuum filter pump was then used for filtration, and the solvent was concentrated using a rotary evaporator to remove excess methanol. The extract paste obtained was stored in a specimen jar at 4 degrees Celsius and is then used in chemical analysis. The chemical analysis performed was the saponins test, also known as the foam test, ferric chloride test for phenolic and tannins, Dragendorf's test for alkaloid, alkyne reagent test for flavonoid, and lastly, Salkowski test for terpenoids.
This is the morphological traits for the sample. This is the result of the phytochemical test.
Next, we will be discussing the result of our research. The first is on the morphometric study. For both the plant, we only do morphometric study on the leaf only. Next, the area for the first plant is classified as macrophyll, while the other one is mesophyll. For the first plant, the width is classified as a thin and membranous, while the other one is moderately coriaceous with coriaceous tendency. The shape for the first plant is lanceolate, meaning it's long, narrow, leaf tapering to a pointed tip, while the other one is oval because the broad leaf with rounded margin and widest at the center. Next is the color. Both show the color green, meaning it chlorophyll reflect green wavelength while absorb light blue for photosynthesis. Next, the venation for the first plant is pinnate, meaning it one midrib with lateral vein, while the other one is reticulate pinnate, one midrib with vein formation, a net-like pattern. Next is the stem. The stem color for the first plant is green and non-wood, meaning it's photosynthetic and flexible, while the other one is brown woody stem. It lacks chlorophyll, hard, rigid to provide mechanical support. For the stomata density, for the adaxial, for the first plant and the second plant, both has low stomata density. But for the abaxial, for the first plant, it has a high stomata density, while the other one just had a moderate stomatal density. Finally, we will be discussing the phytochemical compound found in our plant. First, for Andrographis paniculata, all compounds were found except for terpenoid, while for Melicope ptelefolia, all were found except for alkaloid. First, our phytochemical compound is flavonoid. It's a compound that binds tightly to the cause of inflammation in the body and will also trigger cancer cell death through gene regulation. Second and third, phenolic and tannin. It provides antioxidant and antibacterial properties against pathogen. Also, phenolic will fight bacteria and neutralize free radical. The first one is alkaloid. Alkaloid provide mid pain relieving and blood pressure lowering, and terpenoid and alkaloid will support in reducing infection. Lastly, saponin. Saponin assists with oxidative stress and will also fight microbes in the gut or in skin.
To sum up, this study showed that both Melicope ptelefolia and Andrographis paniculata contains saponins, tannins, flavonoids, and phenolic compounds. These compounds likely explain the plant's properties like antibacterial, anti-inflammatory, and antioxidant effects. Differences between the two plants were distinguished. Terpenoids were weak or absent in Melicope ptelefolia, while alkaloids weren't detected in Andrographis paniculata. This could be because the compounds are present in very low amounts or due to the limits in the extraction process. The findings highlight interspecies variation in secondary metabolites between the Rutaceae and Acanthaceae families. Overall, both plants demonstrate strong potential as source of bioactive compounds for future natural product-based drug development.